In this video, we'll look at the Antibody and Nanobody Dashboard data functions in ChemCharts. The Antibody and Nanobody Dashboard data functions provide workflows that bring together multiple other data functions automatically into a single pipeline. Starting with just the protein sequences for antibodies or nanobodies, structure predictions are generated, sequences are assigned IMGT numbering, relative accessible surface area or RASA is calculated, sequence liabilities are evaluated using sequence and structural information, and developability metrics are calculated. All of these results are combined into a single dashboard visualization, providing a 3D view of the structure, a table with all calculated metrics, and a chart with acceptance ranges for the metrics illustrated. For this example, I have a table with 242 antibody sequences from the paper by Revvity et al. From the settings dialog for the Antibody Dashboard data function, select the data table and the column containing concatenated heavy and light chain antibody protein sequences, and a column containing IDs for each row. If multiple structure prediction models are available, choose which to use for structure prediction. For A-Bodybuilder version 2, select whether to retain only the best or the most representative structure based on RMSD differences in backbone atoms, or all four structures. Predicted structures will be labeled with the model name and the submodel numbers that generated them in the final table. Rows used for prediction can also be limited to only the selected or filtered rows. When the data function is finished, click on the Configure Visuals icon in the data function results pane to create the Antibody Dashboard. The Antibody Dashboard is composed of three visuals: a table containing the calculated results at the lower right, the biomolecule viewer at the left, and a multi-parameter optimization chart, or MPO chart, at the upper right. Looking at the table, the first two columns are fixed in place and contain the ID for the antibody sequence, and a sequence column showing the sequence annotated with antibody numbering, relative accessible surface area, and sequence liabilities. Data columns are grouped into categories. Sequence liability columns have true or false values for each liability, and cells are color-coded red when that liability is found. Since structures were predicted in this case, only those residues categorized as solvent exposed are considered for sequence liabilities. Developability metric columns consist of 5 developability metrics derived from the paper by Revvity et al. Cells are color-coded red, yellow, and green for unacceptable, marginal, and acceptable values based on the publication. 2 additional columns provide counts of red and yellow alerts for each sequence. Cells are colored darker yellow or darker red for larger numbers of alerts. Lengths of each CDR region based on IMGT CDR definitions are also provided. Additional columns contain the predicted structure, original sequence, modeled heavy and light chain sequences, and the model details. The left pane of the dashboard has the biomolecule viewer, and when a row or multiple rows are selected from the table, the predicted structures for those antibodies are shown. The sequence pane below the 3D structure viewer may need to have the rows expanded to see all of the annotations, sequence numbering, RASA, and sequence liabilities. Clicking on a residue in the sequence pane will zoom to that residue in the structure viewer and highlight it. In the upper right of the dashboard is a multi-parameter optimization chart with each developability metric on the x-axis and multiple y-axes showing the ranges of values for the metrics. Red regions indicate values outside of acceptable ranges defined in the Revvity paper and adjusted for calculations done in ChemCharts. Yellow regions are marginally acceptable, and green regions indicate acceptable values. The MPO plot and the table are interactive. The gray lines correspond to sequences in the selected rows of the table. Hovering over a line in the chart will highlight that row in the table. Likewise, hovering over a row in the table will highlight the corresponding line in the MPO plot. On each vertical axis, a distribution plot of the values for that metric is shown. The left side shows a histogram and the right shows a violin plot. The Nanobody Dashboard works similarly to the Antibody Dashboard. From the Data Function Settings dialog, select the data table and column containing nanobody protein sequences and an ID column. If multiple predictive models are available, choose which models to use. For Nanobody Builder version 2, choose whether to retain the most representative structure based on RMSD of the backbone, or structures from all models. Predicted structures will be labeled by which model and submodel was used to generate it. Finally, choose how to limit which rows are used for predictions. Once the data function has finished, click Configure Visuals to create the Nanobody Dashboard. Like the Antibody Dashboard, the Nanobody Dashboard contains a 3D structure viewer with a linked sequence pane, a table of calculated values, and an MPO plot. The data table is also similar to the Antibody Dashboard with fixed ID and annotated sequence columns. Sequence liability columns contain true or false values for each liability, and cells are color-coded red if the liability is found. The same developability metrics are calculated for nanobodies as were calculated for antibodies, except for interchain charge symmetry, since there is only one chain. Also, only heavy chain CDR lengths are computed. The cells in the table are not colored, since no acceptance cutoffs are currently defined for nanobodies. These will be updated as more information becomes available. Predicted structures, original sequences, and model details are also provided. Structures are displayed for selected rows in the table, and the sequence and structure panes in the biomolecule viewer are linked, allowing selection and visualization of individual residues. The MPO plot shows distributions for the 4 calculated developability metrics and is responsive to hovering and selection in the table. As with the table, no acceptance regions are illustrated due to a lack of data at this time. As newer models and metrics become available, these tools will be updated to include them. Thanks for watching, and check out our other ChemCharts videos.